ease into the quiet, let your shoulders soften, and let the night take the weight from you. Tonight, we'll drift softly through the quiet science of why sleep calls you home each night. Should this help you fall asleep, subscribe and I'll have another for you tomorrow. Deep inside the brain, in the narrow fluid space between one neuron and the next, something small is released. It has no color. It moves no more than a few micrometers before it reaches the surface of the next cell and settles there. It is quieter than a breath, and yet across the full length of a waking day, the slow accumulation of this one small molecule will become the most faithful record the brain has ever kept of its own effort. The neuron that released it has been burning since before the first light of morning, not burning the way a fire burns, with heat and visible light and something being consumed, but burning the way a working cell burns, in the deep chemical sense, pulling energy from stored compounds and releasing it in the form of electrical signals, molecular cascades, the constant maintenance of membrane gradients that keep a cell alive and able to fire. Every hour that passes, every signal sent, every synapse maintained, costs something. The currency the brain runs on is a molecule called adenosine triphosphate, and when that currency is spent, when the cell has pulled the last usable phosphate from the chain, what remains is adenosine itself, not destroyed, not consumed, released, drifting outward from the cell into the narrow extracellular fluid, where it joins all the other adenosine molecules released by all the other neurons that have also been burning since morning. Adenosine is small. Its molecular formula is 10 carbons, 13 hydrogens, 5 nitrogens, 4 oxygens, a purine ring structure with a ribose sugar attached, the same core that forms the heart of the energy molecule from which it came. If adenosine triphosphate is a key that opens the door to cellular work, then adenosine itself is what that key becomes after the locks have all been turned, the same shape at its center, the same molecular identity, but stripped of the phosphate chain that gave it power. It no longer opens anything. What it does instead, with the same quiet precision, is close. It closes because it fits. Out in the extracellular fluid, adenosine finds receptors, proteins set into the outer membranes of neurons, shaped exactly to receive it. These are the A1 receptors, and they are distributed across the brain with a particular density in the places that matter most for waking: the cortex, the hippocampus, and the basal forebrain. When adenosine docks at an A1 receptor, a cascade begins. A channel opens in the neuron's membrane, a potassium channel, specifically an inwardly rectifying type, and through that channel, potassium ions move. The movement of those ions shifts the electrical charge across the membrane in a particular direction. The cell becomes slightly less likely to fire. It has not stopped. It has not been damaged or removed. It has simply been dimmed, the way a lamp dims when its voltage drops by a fraction, still present, still functional, but producing a little less light than it did before. That dimming happens across a span of time that is almost imperceptible from moment to moment. In the first hour of waking, the adenosine released by the brain's neurons has barely begun to gather. The receptors are largely unoccupied. The channels are mostly closed. The cells are firing with the full energy of a rested brain. There is no sensation of anything building. There is only the ordinary clean alertness of a morning that has not yet carried any weight. But the neurons keep firing, and the ATP keeps being spent, and the adenosine keeps being released. One molecule at a time by billions of cells, across every region of a brain that is doing the continuous metabolic work of being awake and attentive and responsive to the world. The concentration in the extracellular fluid rises not in any detectable instant, but in the way that water rises behind a dam, smoothly, by a small increment with every passing hour, the change invisible in any given minute, but unmistakable across the arc of an afternoon. The particular region where this accumulation carries the most weight is a cluster of cells deep in the forebrain, tucked just behind and beneath the cortex, in a zone that encompasses structures with old anatomical names, the nucleus basalis of Meinert, the substantia innominata. Together, these form the basal forebrain, and it is here that the relationship between adenosine and sleep is most direct, most concentrated, most consequential. In the late 1990s, researchers used fine glass probes, microdialysis catheters, thin enough to sample the chemistry of a specific small region of the brain without grossly disturbing it, to measure the actual adenosine concentration in the basal forebrain of cats across the full cycle of waking and sleeping. What they found was that adenosine in this region rose steadily across 6 hours of waking and fell steadily across the hours of subsequent sleep. The relationship was clean and direct. The longer the animal had been awake, the higher the adenosine concentration. The more it slept, the lower the concentration fell. Not a metaphor, not a model, a measured chemical tide, rising and falling with the rhythm of the animal's consciousness. That tide, rising by small fractions of a micromole through the morning and into the afternoon, reaches the neurons of the basal forebrain and begins to silence them. These are cholinergic neurons, cells that release the neurotransmitter acetylcholine, and they are among the most important drivers of cortical arousal in the entire brain. Their axons reach upward into the cortex and sustain it in the state of alert, activated wakefulness that makes focused attention possible. When they fire, the cortex hums with activity. When they grow quiet, the cortex loses some of the electrical signature of wakefulness. It begins to soften at the edges, to produce slightly slower oscillations, to drift in the direction of the state it will eventually enter. In the early hours of a morning, these cholinergic neurons fire with a vigor that is partly a consequence of their own intrinsic properties, and partly a consequence of the fact that adenosine has been cleared during sleep, and their own 1 receptors are relatively unoccupied. The Gurk channels across their membranes are mostly closed. Their resting membrane potential sits close to the threshold for firing. The smallest input is enough to push them over, and so they fire readily, repeatedly, sustaining the cortex in a state of broad, diffuse alertness. Picture a single one of these neurons in the early morning. It is a medium-sized cell, irregularly shaped, with a long axon extending upward into the cortex and dendrites extending outward to receive inputs from neighboring structures. Adenosine drifts past it in small concentrations. A few molecules bind to 1 receptors on its surface, open a few Gurk channels, and the cell accommodates its membrane potential shifts slightly, but remains well above the threshold for inhibition. The cell fires. The acetylcholine it releases reaches the cortex stays awake. Now picture the same cell 6 hours later, in the mid-afternoon. The extracellular adenosine concentration around it has roughly doubled. More VANA1 receptors on its surface are occupied. More GR channels have been opened by the receptor binding. The steady outward flow of potassium ions through those channels is holding the membrane potential a few millivolts more negative than it was this morning. The cell still fires. It is not dead, not suppressed to silence, but it fires a little less readily. The threshold it must overcome has risen by a small increment. The inputs that were enough to trigger it at 8 in the morning are no longer quite sufficient at 2 in the afternoon. Some signals that would have driven it to fire now fail to do so. The acetylcholine that reaches the cortex is a little less frequent, a little less sustained, and the cortex, sensing that diminished input, softens its own electrical signature in response. This is not a failure. This is a record. Every unit of adenosine in that extracellular fluid represents a spent unit of ATP, and every spent unit of ATP represents work that the brain has done. The chemistry of exhaustion is also the chemistry of effort. The molecule that is now dimming the cholinergic neurons of the basal forebrain is the same molecule that was powering every thought, every perception, every maintained memory, and sustained attention of the waking day. The accumulation is, in a precise molecular sense, a tally. There is a second receptor type involved in this process, and it works at a different level. The A2A receptor, concentrated in a region of the forebrain called the nucleus accumbens shell, sits within the ventral striatum, a structure more commonly associated with reward and motivation than with sleep. But experiments in mice in which the gene encoding the A2A receptor was specifically deleted revealed something about the specific role this receptor plays. Without it, adenosine still accumulates. The A1 receptors are still occupied, and the neurons of the basal forebrain still feel the growing inhibitory pressure. But the transition into sleep, the actual behavioral act of lying down, the shift from waking to sleep onset, is impaired. The animal stays awake longer despite the homeostatic pressure it has accumulated. What this suggests is that the two receptor types are not doing the same job. The A1 receptor, distributed broadly across the cortex and basal forebrain, is doing the slow, continuous work of dimming and suppressing, accumulating the molecular record of wakefulness, reducing neural excitability, hour by hour, building the pressure that will eventually demand sleep. The A2A receptor, concentrated in its particular striatal location, is doing something more like triggering, converting the accumulated pressure into the actual behavioral act of sleep onset, translating a continuous molecular gradient into a discrete state change. The one measures, the other acts on the measurement. Both are responding to the same molecule. That molecule is adenosine, drifting outward from spent neurons, finding the receptors that were built to receive it, carrying in its simple structure the full accumulated weight of the day's metabolic effort. Adenosine does not travel far. There is no blood circulation carrying it across the brain, no long-range signaling pathway that would allow adenosine released in the visual cortex to accumulate with adenosine released in the motor cortex in some shared pool. The concentration gradients are local. They build in the extracellular space around the neurons that produce them, and the receptors that respond to them are on the nearby neurons that will be most affected by the functional state of those regions. This means that the adenosine record is not a single global counter, but a distributed one, a map of local metabolic effort, built up region by region, each part of the brain carrying its own small chemical trace of the work it has been doing. The basal forebrain is not the only place where this happens. It is the place where it matters most for sleep behavior, where the cells whose silencing has the greatest consequence for the level of cortical activation reside. But across the whole brain, in the fluid spaces between billions of cells, the same quiet chemistry is running. Think of it this way. A single neuron fires. It fires again. It fires across an hour of concentrated attention, hundreds of times, drawing down its local ATP reserves and releasing adenosine into the narrow space around it. That adenosine binds to a 1 receptors on the neuron's own surface, a feedback mechanism, the cell measuring its own depletion, and on neighboring cells, diffusing outward by fractions of a millimeter before being broken down by enzymes or transported back into cells for recycling. The local adenosine concentration peaks and partially clears, peaks again with the next bout of activity, never fully returning to its baseline as long as waking continues, building fractionally higher with each cycle of exertion and partial recovery. This is happening in thousands of cortical columns simultaneously. It is happening in the hippocampus with every memory-forming process. It is happening in the basal forebrain with every cholinergic firing that sustains cortical arousal. It is happening most of all in the regions that are working hardest, the ones sustaining the specific cognitive load of the current moment, the ones that have been active longest without rest. Adenosine does not care about the content of the work. It does not distinguish between thinking a complex thought and maintaining a simple motor rhythm. It accumulates wherever ATP is spent, and it accumulates more in the regions that have spent more. By mid-afternoon, the brain has been spending for 8, 9, 10 hours. The adenosine in the basal forebrain has climbed to a concentration that is measurably higher than it was at waking. The A1 receptors on the cholinergic neurons are more occupied than they were this morning. More GERK channels are open across more membranes. The resting electrical state of these cells has shifted, millivolt by millivolt, in the direction of inhibition. They have not stopped firing, but the fraction of inputs they fail to respond to has grown. The acetylcholine reaching the cortex has thinned, and the cortex, unsupported by the full cholinergic drive of the morning, has begun to produce the faint early signatures of something other than full wakefulness. A slight increase in slow oscillations, a marginal rise in the theta range, a softening at the edges of attention. This can be measured. Place fine wire electrodes on the scalp and record the electrical activity of the cortex through an afternoon of sustained wakefulness. There are changes, not dramatic, not sufficient to produce sleep on their own, not anything that would be visible to an outside observer, but real, measurable changes in the frequency distribution of the brain's electrical output. The delta and theta bands creep upward in power. The high-frequency activity that marks alert, engaged wakefulness reduces by fractions. The record on the screen is slowly changing its character, and the molecule responsible for that change is still accumulating, one spent ATP molecule at a time, in the fluid spaces between cells that have been working since morning. The structural resemblance between adenosine and the molecule it comes from is worth sitting with for a moment. Adenosine triphosphate, the energy currency of the cell, is built around an adenosine core. The adenosine molecule that will eventually inhibit the basal forebrain is already present, structurally, in every molecule of ATP that powers the neuron's activity. The phosphate groups attached to it are what make it an energy carrier. When they are removed, first one to produce ADP, then another to produce AMP, then the last by an enzyme called 5-nitronucleotidase, what is left is adenosine itself, unchanged at its core, newly free to diffuse outward and bind to the receptors that were built to receive it. The same molecule that powered the work becomes the signal that the work has been done. The fuel and the fatigue share a structure. They are, in the deepest sense, the same thing at different stages. There is something in this that the nervous system has been using for a very long time. Adenosine-based signaling is ancient, found in organisms far simpler than vertebrates, conserved across hundreds of millions of years of evolution because the relationship between metabolic work and adenosine release is one of the most fundamental chemical relationships in cellular biology. Every living cell that runs on ATP leaves adenosine in its wake. The brain has built a sleepiness detector out of this fact, converting a universal consequence of metabolism into a specific behavioral signal. The system requires no special energy sensor, no dedicated fatigue measuring circuit, no separate monitoring system that has to be maintained alongside the circuit it monitors. It uses the byproduct itself. By the time evening approaches, 16 hours into a waking day, the basal forebrain bathed in a concentration of adenosine that has been building since dawn. The cholinergic neurons are producing noticeably less output than they were this morning. Not silence, not cessation, but a measurable functional reduction in the drive they are sending upward into the cortex. The cortex, receiving that reduced drive, is sustaining wakefulness with a little more effort and a little less ease. The A1 receptors are heavily occupied. The Gurk channels are open across membrane after membrane. The electrical balance inside these cells has shifted toward the threshold of inhibition, the membrane potential sitting deeper, the firing rate reduced, the probability that any given input will produce a response lower than it was 8 hours ago. The experience of this from the inside is familiar. The slight heaviness behind the eyes, the incremental reduction in the sharpness of sustained attention, the way complex tasks that were easy in the morning require a fraction more effort by late afternoon, the brief, recoverable lapses in concentration that appear and then close. None of these are dramatic events. They are the felt consequence of a molecular tide that has been rising across the entire day in fluid spaces 1,000th the width of a human hair, through mechanisms so small and so precise that the instruments to measure them were invented only in the last few decades of human history. This is what sleepiness is, at its root. Not a vague mood, not the absence of motivation, not a failure of discipline or effort. It is a specific molecular state, an extracellular adenosine concentration that has reached a level sufficient to occupy enough dead, one receptors, open enough Gurk channels, and shift enough membrane potentials that the wake-promoting neurons of the basal forebrain are running measurably quieter than they were at the start of the day. It is the chemical consequence of metabolic work, and it is faithful. It tracks the work with a precision that no conscious record could match, accumulating without pause or error across every hour of exertion, carrying in the concentration of one small molecule the full, unabridged history of the day's effort. The neurons of the basal forebrain have been producing acetylcholine since the first minutes of waking. They sent it upward into the cortex all morning, sustaining the clean, sharp attentiveness of a rested brain. They sent it through the middle hours of the day, the drive still strong, but beginning to feel the first fractional pressure of the adenosine rising around them. They have been sending it all afternoon, a little less readily than this morning, the signal growing marginally thinner with each hour that passes, the Gurk channels opening one by one as more receptors are occupied, the membrane potential settling a little deeper with each passing cycle of inhibition. By the time the last light has gone from the sky and the long day has finished its work, these neurons are quieter than they have been since before waking, not silent, not yet, but substantially reduced from the vigorous early morning firing that held the cortex in its most alert state. The adenosine that surrounds them in the extracellular fluid of the basal forebrain, pressed close to their surfaces, occupying their A1 receptors, holding their potassium channels open, is not a chemical intruder. It is a record. Every molecule of it came from spent ATP, from work done, from signals sent, from the ordinary metabolic effort of being a living, thinking brain across the full arc of one more day. Sleep, When it comes, we'll begin to clear that record. Adenosine will be reabsorbed and recycled. The concentrations will fall. The A1 receptors will be progressively vacated. The GERK channels will close, one by one, as the extracellular space is cleared. The membrane potentials will return toward their well-rested morning values, and the cholinergic neurons of the basal forebrain, receiving less and less adenosine pressure as the night deepens, will recover the firing threshold they had at the start of the day, ready to sustain the cortex in alertness when morning comes again. But that recovery belongs to the night ahead. In the last hour of this evening, the process of accumulation is still running. The basal forebrain is still bathed in its slow tide of adenosine. The cholinergic neurons are still firing, quieter now, their output thinned, the cortex they support running on reduced drive. And the sleepiness that has been building since the first hour of the morning is exactly what it was always going to be, the precise molecular consequence of a brain that has been working faithfully since dawn, leaving its record in the fluid between cells, one spent ATP molecule at a time. There is nothing wrong with this. There has never been anything wrong with this. The dimming of the basal forebrain's cholinergic neurons at the end of a long day is not deterioration, it is completion. The record is full. The molecule that was the fuel is now the signal, and the signal, rising quietly through the evening in the narrow fluid space between cells, says only what it has always said, that the work is done and the time for rest has arrived. In a small office in Zurich, in the middle of an ordinary afternoon, a researcher sat with a stack of sleep records spread across his desk, EEG tracings printed on long paper, each one a day's worth of slow oscillation, compressed into something that could be held in two hands. Alexander Borbeli had been looking at sleep for years. He had measured its depth and its timing, its chemistry and its electrical texture, and one afternoon, working through those records, he found himself facing something that a single mechanism could not explain. The problem was this: sleep did not simply arrive when enough fatigue had gathered. If that were the whole story, then the deepest sleep would always follow the longest waking, and its timing would slide freely across the clock, arriving wherever the hours of effort pointed. But sleep did not behave that way. It arrived at particular times. It resisted displacement. Its depth and its architecture, the proportions of slow dreaming and fast, the distribution of delta waves across the early hours of the night, these were shaped by something that did not care how long a person had been awake. Something else was running, something with its own curve, its own period, its own indifference to the hours of accumulated effort. Borbeli looked at the data for a long time, and then he reached for the only explanation that fit. There were not one but two separate processes shaping when sleep came, how deep it ran, and when it released its hold on the waking day. He called them Process S and Process C. The names are plain, almost deliberately so, letters from the words homeostatic and circadian, reduced to their initial sounds, like two variables in an equation waiting to be solved. But inside those plain names lived one of the most complete pictures of a biological system that sleep research had produced. Borbeli published the model in 1982 in a journal called Human Neurobiology, and the architecture he described that year is still the frame that most sleep research is built upon. Not because it was simple, because it was true. Process is the one that counts the hours. It rises from the moment of waking, not in a straight line, but in a curve, exponential in its shape, each waking hour adding slightly less to the load than the hour before, the way a vessel fills fastest when it is empty and slows as it approaches its limit. The accumulation is real, and it is molecular, a slow tide of biochemical signal building in the narrow fluid channels between nerve cells, most densely in the region of the brain where wakefulness is most actively maintained. It is the record of effort. The brain keeps the score of every hour it has been running, and it writes that score in chemistry. Through the morning, the curve rises. Through the afternoon, it continues its slow climb, and the body begins to feel its weight in ways that are subtle at first. A slight heaviness behind the eyes, a mild reluctance where there was once readiness. Nothing dramatic, nothing that could not be managed. The record of the waking hours is building, as it always does, in the same patient upward arc it traces every day, toward the threshold where sleep becomes not merely possible, but close to irresistible. Process C is something else entirely. It does not count hours. It does not measure effort or metabolic cost. It oscillates, moving through its cycle at a period close to 24 hours, rising through the day and falling through the night with a steadiness that is wholly indifferent to what the body is doing. Whether the body is running or resting, working or still, sleeping or awake, Process C continues along its curve, cresting at its own appointed time, falling at its own pace, governed by a biological clock deep in the brain that sets its period not by fatigue, but by gene expression cycling inside individual neurons. Process C is not a record, it is a rhythm, and that distinction between the record and the rhythm is the entire difference between the two processes, the conceptual gap that makes the model necessary and makes the feeling of sleepiness something more intricate than mere accumulation. Picture process drawn as a line rising across a day. It begins low at waking before the first hour of the morning has passed. It climbs through the late morning hours with the steady patience of water filling a very deep vessel. By midday, it has risen noticeably, though the circadian alerting signal is still strong enough to hold it comfortably in check. Through the early afternoon, it continues upward, and the body registers the pressure in small ways, a faint thickening of thought, a drop in the precision of fine attention, the sensation that the world has become very slightly slower and more distant. And the curve is gradual, and its slope does not change dramatically from one hour to the next. It is the patient accumulation of effort, written in chemistry, rising toward a threshold that by late evening it will reach. Now picture process C as a different kind of line entirely, not rising but oscillating, a long smooth wave that moves through its own period without reference to any clock a person carries. Through the morning it climbs steadily, the circadian alerting signal growing stronger through the early waking hours, countering the rising weight of process S and holding wakefulness clear and relatively effortless. Through midday, it sustains this alerting force. Through the early afternoon, there is a secondary dip, a slight valley in the circadian curve that the biology of the afternoon inscribes, regardless of whether food has been eaten or a long morning spent at rest. And then, from the middle of the afternoon into the early evening, the circadian alerting signal rises again to its highest point of the entire day, a paradoxical crest that arrives at precisely the moment when process S has been climbing for 16 or 17 hours and might be expected to overwhelm everything else. This is the arrangement that makes the evening feel different from the morning. The morning's alertness is helped by a process S that is low, recently discharged by the night's sleep, and by a circadian signal that is climbing. By early evening, process S is high. Hours of wakefulness have built the pressure to its daily peak, but the circadian signal is also near its peak, pushing back against that pressure, holding the door open. The two processes are working in opposition, and the net result is a period of genuine, measurable alertness in the early evening that is not diminished by the hours of waking behind it. Process C is holding back the tide. This period has a name in the sleep literature. Researchers call it the wake maintenance zone. It is not a metaphor. It is a measurable physiological phenomenon, a window of roughly 1 to 2 hours in the early evening when the likelihood of falling asleep is paradoxically lower than it was in the early afternoon, despite the full load of a day's accumulated sleep pressure pressing from the other side. Charles Czeisler and his colleagues at Harvard quantified it with precision, using a protocol designed to pull the two processes apart and examine each in isolation. They placed study participants on a 28-hour artificial day. Cycles of waking and sleep extended far beyond what the natural circadian clock expects, so that over many days, the body's internal clock drifted slowly out of alignment with the participants' actual sleep and wake schedule. With the two processes running at different frequencies, no longer locked together, the researchers could measure each variable independently, mapping the circadian and homeostatic components of sleepiness across the day without the confounding of one by the other. What they found was a circadian alerting signal whose late evening peak was sharp and real, a genuine biological push toward wakefulness in the early evening that had nothing to do with the hours of effort preceding it. The wake maintenance zone was not a felt impression, it was data. Inside each individual's biology, the wake maintenance zone represents a kind of conversation between two systems that never directly speak. Process has no awareness of the circadian clock. It accumulates adenosine through neuronal activity and does not check the hour. Process C cycles through its own period, driven by molecular oscillation inside hypothalamic neurons, indifferent to the metabolic record being kept elsewhere. And yet the two processes interact in their combined effect on wakefulness, not through direct signaling, but through their opposition in the circuits that maintain arousal. When both are high, wakefulness is maintained against the pressure of sleep. When process C begins its evening fall, the opposition dissolves, and the accumulated weight of process S is suddenly unopposed. This is the moment that matters most in the arc of a waking day. It does not arrive all at once. The circadian alerting signal does not cut off at a precise hour, leaving the day ended. It falls gradually over the course of an hour or more in the late evening, but that fall is coupled to something else, another signal beginning its own rise at nearly the same moment, a hormonal tide that starts in a small gland at the back of the brain and spreads across the body in the bloodstream, reinforcing the circadian fall with chemistry the blood can carry. The two processes releasing together in the late evening, the alerting signal dropping, its chemical counterpart rising, create a convergence that is different in quality from either process alone. The pressure of process S has been building toward this moment for the entire day. The circadian clock has been moving toward it at its own independent pace. When the two arrive at the same point simultaneously, the combined effect is the sensation of genuine structural sleepiness, not merely tiredness, not a preference for rest, but a biological condition in which sleep becomes the natural continuation of the moment, the thing the body has been moving toward since the morning. Bohr-Bailey captured this in the mathematics of the model. Process S follows an exponential charging curve during waking hours, rising toward an upper asymptote, a ceiling defined by the system's own parameters, and an exponential discharge curve during sleep, falling toward a lower asymptote as the night's sleep repays the accumulated debt. The curves were not drawn by hand or intuited from description. They were derived from empirical data, from measurements of slow-wave EEG activity during sleep and during recovery after deprivation. Slow-wave activity, the dense, high-amplitude oscillations in the lowest frequency range of the sleeping brain, serves as the marker of process S, its visible correlate in the electrical record of the brain. When sleep pressure is high, slow-wave activity in the first few hours of the night is intense and front-loaded, the brain repaying what it owes in the deepest currency it has. When sleep pressure is low, after a nap, after a night of unusually long sleep, slow-wave activity is reduced, the curve having already discharged part of its load before the main night begins. Working with Peter Ackerman, Borbély encoded these dynamics in a set of differential equations, mathematical expressions that describe how the two variables change across time and interact in their combined effect on sleep timing. The equations have parameters, numbers that set the height and shape of the curves, and these parameters were derived from the slow-wave rebound data, matched to what human sleepers actually showed after nights of deprivation and recovery. The model predicted when people would fall asleep, how long they would stay asleep, and how the architecture of that sleep, its proportions of slow and fast, its distribution of delta waves across the night, would be shaped by the preceding waking period. It predicted things that had not yet been measured, and when those things were measured, the model held. There is something quietly striking about the fact that the two processes have such different origins. Process S grows from the work the brain does, from the metabolic activity of neurons firing and sustaining the electrochemical gradients that allow them to fire again, from the slow accumulation of a purine nucleoside that is, in a sense, the chemical trace of thought itself. Rising in the narrow fluid spaces between cells, with every hour the brain runs, it is local and molecular and directly tied to the brain's own effort. Process C, by contrast, was generated by a clock so small it could rest on a grain of rice, a paired structure of roughly 20,000 neurons per side, sitting directly above the point where the two optic nerves cross, deriving its period not from metabolic activity, but from a self-referential loop of gene expression, proteins inhibiting the very genes that made them, cycling with a period close to 24 hours that was set not by anything the organism does, but by the biochemistry it inherited. The two processes are about as different in origin as two biological mechanisms could be, and yet their interaction, timed as it is across the arc of a day, produces the most ordinary and universal of human experiences. The gradual settling into evening, the growing weight of the hours behind, the moment when the body stops holding the day open and allows the night to begin. The interaction between the two processes also explains things that seem paradoxical from the outside. Why does a person who has been awake for 20 hours sometimes feel a second wind in the early evening, a brief return of clarity that arrives without any sleep to account for it? Because process C is near its nightly peak, and its alerting force is strong enough to temporarily override the heavy accumulation of process S. Why does the early afternoon feel heavier than the late morning, even though process S has been accumulating continuously since waking? Because the circadian alerting curve has a secondary valley in the early afternoon, a genuine dip inscribed by the biology of the clock that produces a brief reduction in the alerting force countering the homeostatic load. The afternoon dip is real and measurable, detectable in core body temperature, in the EEG record of a resting brain, in performance on tasks that require sustained attention. It is not explained by lunch. It was there before lunch was a habit. The two-process model also makes sense of what happens when the two processes are forcibly separated, when sleep is delayed far into the night, or taken at the wrong biological hour, or fragmented across a stretch of time that the circadian clock was not built to accommodate. In shift workers, the two processes often run in opposition rather than in concert. Process S demanding sleep while Process C is generating its daytime alerting signal, or Process C demanding sleep while Process S remains partially discharged from a daytime nap taken in biological night. The result is a sustained misalignment between the two systems, a condition in which neither process can do what it does best, because the other is running against it rather than with it. The sleepiness that accumulates in this condition is qualitatively different from the comfortable evening settling of the two processes in alignment. It has an edge to it, a restlessness that coexists with exhaustion, a wakefulness that persists when sleep is needed, and a heaviness that arrives when waking is required. The two-process model gives this a structure, not just a description of what it feels like, but a diagram of why. Understanding the two-process model changes the texture of what it means to feel tired. The heaviness that gathers through the afternoon is not disorder. It is process doing exactly what it should, accumulating the record of the day's metabolic effort, holding it in chemical form, waiting for the circadian signal to release it into sleep. The clarity of an early morning after full sleep is not simply the absence of effort. It is the combination of a fully discharged process and a rising circadian alerting signal, both working in the same direction, both pushing toward wakefulness with their combined force. The long waking hours and the quiet of the night are not opposite ends of a simple continuum. They are the two arcs of a process that has been running inside the body without interruption, without needing instruction, without ever losing the thread of what it was counting. There is a quality to the two-process framework that is different from most explanations of why the body does what it does. Most such explanations are stories about mechanisms, this protein, that receptor, this cascade of signals. The two-process model is something else. It is a description of the body's relationship with time, not just time in the sense of hours elapsed, which process tracks, but time in the deeper sense of period, the way the circadian clock carries inside it, a version of the day that it would cycle through even in the absence of light, even in the absence of all external cues, running its 24-hour program from inside the chemistry of a tiny cluster of neurons that need nothing from the outside world to keep the count. The body knows what time it is. It has always known. And the feeling of sleepiness growing through the late evening is, among other things, the feeling of two different kinds of knowing coinciding, the record of accumulated effort meeting the signal of the hour and together pointing in the same direction. Borbelli proposed the model because the data required it. No single process could account for both the timing of sleep and the depth of it, both when it came and how it felt when it arrived. The gap between those two things demanded a second variable, an independent curve running alongside the first, and in the gap between those two curves, in the space where they oppose and converge and finally align, lives most of what is felt across the ordinary passage of a waking day. The slight heaviness of mid-morning as process S builds against a still strong circadian signal. The valley of the early afternoon when the circadian curve dips and the homeostatic weight briefly gains the upper hand. The surprising clarity of early evening when process C reaches its peak and the two forces balance, briefly with the circadian signal pushing hard against the accumulated load. And then the long gradual fall of the late evening, the circadian alerting signal beginning its retreat, the hormonal tide beginning to rise in the bloodstream, the two processes releasing their opposition at last, the record of the day and the rhythm of the body converging. The convergence does not feel mechanical. It never does from the inside. From the inside, it feels like the day becoming heavier in a gentle way, like a light that has been running for hours, not failing, just settling, finding the lower, softer register it always moves toward at this hour. The eyelids carry more weight than they did this morning. The sounds of the room seem slightly more distant. The edges of things are still clear, but there is no urgency in them. Process has built its record through every hour of the day, and that record is exact, written in the slow accumulation of a molecule that the brain itself produced, rising in the fluid between cells, reaching across hours toward the circuits that will finally answer it. And process C has completed another arc of its long independent oscillation, cresting in the early evening and now beginning its fall, pulling the last resistance from the waking state and allowing the accumulated pressure of the day to move the body toward what it has been building toward since morning. The two processes were always running at once. Through the first waking hours, they were pulling in the same direction, both working toward clarity, toward alertness, toward the full engagement of the day. Through the afternoon, they moved into partial opposition, process pushing one way, and process C holding steadily against it. And now, in the late evening, they released together, the alerting signal dropping from its peak, the homeostatic record meeting the falling threshold, the waking state softening towards something else, something the body has been moving toward all day, with the patience of a process that does not hurry and does not wait, only oscillates and accumulates, and finally, at the hour it was always going to arrive at, allows the day to end. The slow fall of the circadian alerting signal is the last thing standing between the waking day and the night. As it drops from its evening peak, the chemical tide of the night continues to rise, spreading through the bloodstream, reaching the circuits that have been holding wakefulness in place through the long afternoon. The arousal networks that have been active since morning grow quieter, not all at once and not suddenly. One by one, the chemical voices of the waking brain reduce their output, norepinephrine dropping from the brainstem, histamine falling silent in the posterior hypothalamus, the stabilizing projections of the lateral hypothalamus loosening their hold on the whole distributed network of wakefulness. The brain is not failing, it is completing. The two processes have brought it to exactly this point, by exactly these means, at very nearly this hour every night since the first time this body learned to sleep. And in the quiet of the late evening, when process has reached its peak and process C has passed through its crest and begun its long descent, the two systems finally point in the same direction again, both of them now pulling towards sleep rather than away from it. The record of the day's effort aligns with the rhythm of the body's deep clock, the record and the rhythm both pointing down toward the dark, toward the slow wave, toward the long patient dissolution of the day. The body has been moving toward this moment since the morning. The two processes have carried it here, one counting every hour of effort, the other holding its independent course through time. And now they meet in the quiet of the late evening, as they were always going to, at the hour the body was always going to release the day and allow at last the night to begin. Two tiny clusters of cells sit at the base of the brain, just above the place where the optic nerves cross. Each one is smaller than a grain of rice. Together they weigh less than a raindrop, and yet inside them, in neurons no wider than a few thousandths of a millimeter, a clock has been running without interruption since before the first memory was formed, ticking not in seconds, but in the slow molecular language of proteins building and dissolving, genes switching on and then gently off, a cycle that completes itself once in every passage of a day. These two nuclei sit at the very center of the brain's floor, cradled in the hypothalamus, directly above the point where the two optic nerves meet and part. Their position is not incidental. They are close enough to the eye's incoming fibers to receive a private signal, not the signal that carries images or color or the shape of a face, but a simpler, older signal, the one that carries only the bare fact of light, whether it is bright or dark outside, whether the day has begun or whether it has ended. This is the only information they need. That structure, those two small paired bodies, together called the suprachiasmatic nucleus, is the origin of the circadian rhythm, not its expression, its origin, the place where the roughly 24-hour cycle of the body begins, in molecular events so small that the instruments capable of measuring them were not built until very recently in human history. What runs inside those cells is a clock in the most literal sense, a self-correcting, self-sustaining oscillation that keeps time not with wheels or crystals, but with the rising and falling concentration of proteins inside individual neurons. Each of those neurons, taken out of the brain entirely, removed from every neighbor and every input, placed alone in a dish of nutrient solution in a dark laboratory room, each one will continue to oscillate. Its gene expression will rise and fall. Its electrical activity will pass through a cycle of roughly 24 hours and then begin again without any cue from outside, without any neighbor to synchronize with, without any knowledge of whether it is day or night beyond the walls. A single cell sustaining a day. The clock is not the network. The clock is in each cell individually, and the network makes it precise. Inside each of those small neurons, a loop of molecular events turns like a wheel. It is not fast. It takes most of a full day to complete one rotation. The loop has two arms, one that drives the clock forward and one that gently brings it back, and the tension between them is what keeps the timing close to 24 hours, generation after generation, species after species, with a fidelity that has been maintained across hundreds of millions of years of evolutionary history. The driving arm begins with two proteins. They are called clock and BML1, and in the early part of the molecular day, they find each other in the cytoplasm of the cell, the fluid interior, dense with machinery, warm and crowded with other molecules going about other business. These two proteins are drawn together. They bind. They form a pair, a stable double unit, and then they move. Together they enter the cell's nucleus, where the DNA is coiled and stored, and there they attach to specific short sequences of genetic code, sequences called E-boxes that sit upstream of the genes they are meant to activate. When clock and BML1 bind those sequences, the genes downstream of them begin to be read. 4 of them in particular, named PER1, PER2, CRY1, and CRY2, begin producing their proteins. The PER proteins and the CRY proteins begin to appear in the cytoplasm in small quantities, then larger ones, building through the morning and into the afternoon of the molecular day, accumulating across hours in a slow and patient rise. That rise is not fast. It cannot be. The clock's period, its roughly 24-hour cycle, depends entirely on how long it takes for those proteins to accumulate to a concentration sufficient to act, and it takes hours. The PER proteins in particular are held in the cytoplasm by a process of phosphorylation, small chemical tags added by enzymes that delay their entry into the nucleus, keeping them waiting in the cell's outer room while the concentration builds. This delay is not a flaw. It is the mechanism. The wait is the clock. Eventually, when enough PER and CRY protein has gathered, the proteins pair with each other. They form complexes, clusters of molecules bound together, and those complexes move into the nucleus. There they do something precise and necessary. They find the clock, BML1 pair still sitting on the epoch sequences, still driving transcription, and they suppress it. They bind to clock and BML1 and silence them. The drive goes quiet. The genes that were being read begin to go still. The production of new PER and CRE proteins slows, then stops. Now the existing PER and CRE proteins begin to be broken down. There are two enzymes responsible for this, one called beta-trel-CP and another called FBXL3, and they mark the PER and CRE proteins for destruction by attaching small molecular tags called ubiquitin. The tagged proteins are recognized by the cell's protein disposal machinery and disassembled. Their concentration falls. The suppression they were providing on CLUC and BML1 lifts slowly as the proteins disappear, and with that lifting, the drive begins again. CLUC and BML1 are free once more to bind the epoxes. PR and CRE genes are activated again. New PER and CRE proteins begin their slow accumulation in the cytoplasm, beginning another long patient wait before the loop turns once more. The full rotation, drive, accumulate, suppress, degrade, release, drive again, takes approximately 24 hours. Not exactly. No biological clock is exact, but the precision is extraordinary for a system made of molecules, and the small imprecision is corrected every day by the signal that comes in from outside, from the light. That correction depends on a pathway that is entirely separate from the one that carries visual information to the cortex. Deep in the retina, alongside the rod and cone cells that detect shape and color, there is a third population of light-sensitive cells. They are fewer in number. They respond more slowly to light. They do not contribute to forming an image of the world. What they do instead, their single dedicated function, is measure the overall brightness of the environment, the ambient luminance of the sky, and report it to the suprachiasmatic nucleus directly. These cells express a photopigment called melanopsin, and their light sensitivity is tuned to short wavelengths, the blue end of the visible spectrum, the wavelength that dominates the morning sky. When light falls on them, they generate a signal. That signal travels along a pathway called the retinohypothalamic tract, a thin, dedicated fiber that runs not toward the visual cortex, but directly to the ventral core of the suprachiasmatic nucleus. The signal arrives there, carrying one piece of information. It is bright, or it is not. The clock receives this and adjusts. When the retinohypothalamic tract delivers light to the SCN's ventral core, the cells there release two chemical signals, glutamate and a compound called PACAP. These signals act on the clock neurons, and in those neurons, they do something specific and rapid. They induce the immediate transcription of the PR genes. Not a slow accumulation, a rapid burst. The clock receives the light signal as a fast push, a forced early entry of PER protein into the feedback loop, and this push shifts the phase of the oscillation. The clock adjusts its time. The direction of the shift depends on the time of the biological day when the light arrives. Light in the early morning, when PER levels are low and rising, advances the clock. It sets the oscillation slightly earlier. Light in the evening, when the molecular day is ending, delays it, pushes the cycle slightly later. Light in the biological middle of the day, when the clock is at a phase where it is relatively insensitive to this kind of input, has little effect at all. This relationship between light timing and clock adjustment has been mapped in careful detail. The result is called a phase response curve, and it shows the clock's sensitivity to light across the full 24-hour cycle, where the same amount of light will do almost nothing and where a brief exposure will shift the oscillation by hours. Morning light is the most powerful daily signal the body has for keeping its internal timing aligned with the external world, and the melanopsin cells in the retina are calibrated precisely to detect it. What all of this means, taken together, is that the body carries inside its hypothalamus a self-correcting timekeeper with a resolution that no external instrument can match for its purpose. It does not drift the way a pendulum clock drifts in changing temperature. When it runs slightly fast or slightly slow, which all biological clocks do, it is corrected the next morning by the light signal from the retina, and the day's timing is restored. There are people in whom one of the clock proteins is altered by a change in their DNA, a single letter different in the genetic sequence of the PER2 gene. In those people, the molecular clock runs faster than normal, the feedback loop turns in somewhat less than 24 hours. The timing of everything shifts. Sleepiness arrives earlier in the evening, sometimes before other people have finished their working day. Waking happens earlier in the morning, sometimes before 4 o'clock, the body certain that day has come when the sky outside is still completely dark. This is not a behavior or a preference or a failure of discipline. It is a molecular clock running at the speed its proteins dictate. The rest of the sleep and waking cycle, the hormones, the alertness, the body temperature, all follows the clock, and so when the clock shifts, everything shifts with it. This condition has a name: familial advanced sleep phase syndrome. In families where this DNA change is inherited, generation after generation, children and parents and grandparents all wake in the early hours, all feel the pull towards sleep in the early evening, all living in a time zone that the rest of the world does not share, driven there by a single change in a single protein. The existence of that condition is one of the clearest demonstrations available that the subjective experience of sleepiness at a particular time of day is not arbitrary. It is written in molecular code. The sensation of the evening closing in, heaviness that begins to settle as the day lengthens, the way the body begins to lean toward rest long before rest is taken. These experiences are downstream effects of a feedback loop running inside neurons no wider than a small fraction of a millimeter, turning at a rate set by the speed at which proteins are built and dismantled, corrected each morning by the arrival of light through the retina. The suprachiasmatic nucleus communicates its timing outward to the rest of the brain and body through multiple channels. Some are electrical. The neurons of the SCN fire in patterns that change across the day, sending rhythmic signals down into the hypothalamus and beyond. Some are chemical. Neuropeptides released by SCN neurons diffuse through the local tissue and act on neighboring structures. And some are hormonal, mediated by pathways that carry the clock's signal to organs and tissues far from the brain itself. The neurons in the SCN's dorsal shell, which make a peptide called arginine vasopressin, fire with a pattern that peaks in the late morning and early afternoon of biological day. The ventral core neurons, which make different signaling molecules called VIP and GRP, receive and process the light input from the retina and distribute timing corrections across the nucleus. The whole structure operates like a small layered community, input arriving at the center, timing maintained at the population level, output flowing outward from the shell. What the SCN does not do is sleep. It remains active across the full 24 hours. Its neurons do not shut down at night. They shift their firing patterns, change their output, mark the passage of biological time, but the clock itself runs continuously, because a clock that stopped would have to be reset rather than corrected and would lose its accumulated precision. The oscillation is the clock. The clock never stops. A single SCN neuron in a dish of warm fluid, far from any sky, far from any signal, will oscillate through one molecular day, and then another, and then another. It will do this for weeks if the conditions are maintained. Its PER proteins will accumulate and be destroyed on a cycle it generates entirely from within. Its membrane potential will rise and fall on a rhythm it does not borrow from anything outside itself. It is a clock that needs no winding. The community of neurons in the intact SCN is more precise than any individual cell alone, because the cells communicate with each other and average out their small individual variations. A single neuron might run a few minutes fast or a few minutes slow on a given day. The network of 20,000 neurons on each side corrects for those individual variations through the signals they share. VIP released from the ventral core, vasopressin from the dorsal shell, a constant low-level chemical conversation that keeps the population of clocks synchronized into one larger, more accurate signal. In the morning, when light from the retina arrives through the retinohypothalamic tract, that signal reaches a system that is already running, already coordinated, already tracking its internal time, and nudges it gently toward alignment with the outside world. The nudge is subtle on most days. The clock's own endogenous period in most people runs only slightly longer than 24 hours. The daily correction needed is small. It takes only a few minutes of morning light to keep a clock that runs at 24 hours and 10 minutes exactly synchronized with a world that rotates in exactly 24. But the clock's sensitivity to that light is not the same at every hour. In the early morning, when biological day is just beginning, the system is maximally responsive to light that advances the phase, that says, "Start now, the day is here, the darkness is behind you." In the evening, the same amount of light pushes in the opposite direction. The clock reads it as a signal that day is ending later than expected, and shifts accordingly, delaying the following morning's biological dawn. This is why the quality of light in the hours before sleep has consequences that reach past the evening and into the next day's timing. The melanopsin cells in the retina, sensitive to short wavelength light, are most strongly activated by the kind of light that comes from a clear morning sky, bright, broad spectrum, rich in the frequencies that their photopigment absorbs best. Indoors, in the evening, in the orange and amber tones of candlelight or incandescent bulbs, these cells are relatively quiet. But in the blue-enriched light of certain artificial sources, lamps designed for efficiency, the glow from a lit screen, they are activated, and the signal they send to the suprachiasmatic nucleus reads as, "Day is still here." The clock receives that signal as it would receive morning light. It shifts, the oscillation moves. What this means for the felt experience of sleepiness is not straightforward to describe without mechanism, but the mechanism is now visible. The circadian component of sleepiness depends on where the SCN clock currently sits in its oscillation. When the clock's output signals are in the phase that promotes waking, when vasopressin release is high, when the SCN's firing patterns are in their daytime configuration, the brain's arousal systems are supported. When the clock moves into the phase that withdraws that support, when firing patterns shift toward their nighttime form, the felt experience of the evening closing in arrives. That experience is not simply tiredness, it is the clock withdrawing its active opposition to sleep. This is worth holding still for a moment. The sense of evening heaviness, the way concentration softens, and the world seems to acquire a gentler texture in the late hours. That experience is partly the absence of something active, not the arrival of something new. Across the day, the SCN has been providing a quiet, steady support to the systems that maintain wakefulness. And as it moves through the second half of its cycle, that support gradually withdraws. The waking state does not fail. It is simply no longer being held open with quite the same insistence. The full circuit, from molecular clock to felt sleepiness, passes through many stages. The loop of pair and cry proteins, turning inside SCN neurons smaller than a grain of sand, generates a timing signal. That signal passes outward through the SCN's network, shaping the firing patterns of the whole nucleus. From there, it reaches the hypothalamus and the wider brainstem, influencing the populations of neurons that sustain wakefulness and those that promote sleep. And eventually, at the end of that long chain of molecular and electrical events, a person lying on a sofa in the late evening feels the day beginning to close around them, the room softening slightly, the effort of staying alert beginning to cost something more than it did at noon. That experience arrives reliably because the clock is reliable. It arrives at roughly the same hour because the clock's period is roughly the same each day. It arrives a little earlier in winter when morning light comes later and the clock's daily correction is timed differently, and a little later in summer and at different hours in people whose molecular clocks run at slightly different speeds. And all of those variations are direct expressions of the molecular mechanism, the same feedback loop of PER and Cre proteins, the same correction arriving each morning from the retina, shifted slightly in its timing by the length of the day or the particular sequence of DNA in the PER2 gene. The retinohypothalamic tract carries no image. It does not contribute to the perception of color or depth or the recognition of a face. It is a dedicated line carrying only one kind of message from one kind of cell in the retina, those slow, blue-sensitive melanopsin-containing cells, to one small target in the hypothalamus. The tract is thin. It is not easily visible to the naked eye. It carries less information than almost any other fiber in the visual system. What it carries is enough: the fact of light, or its absence, the quality of the morning sky, the single raw signal that the clock needs to stay synchronized with a rotating planet, and that has been arriving each morning at the same small cluster of neurons for as long as those cells have existed in any form that resembles the one they hold now. Somewhere in those 20,000 neurons on each side, a total volume smaller than a sesame seed placed at the base of the brain, a protein pair called clock and BMAL1 is completing its daily binding to the EBOX sequences of the Cry genes, setting in motion the accumulation of proteins that will, across the next 12 hours or so, grow in concentration and then return to suppress the very drive that created them. And then that suppression will lift, and the drive will start again, and the clock will continue its turn. The proteins do not know what day it is. They do not know what season, what latitude, what the sky looks like outside the body that contains them. They only know their concentrations, their binding affinities, the kinetics of their phosphorylation and degradation. And yet from those purely local facts, from nothing more than chemistry inside a small warm cell, something emerges that is as regular as the turning of the earth, and nearly as old, and just as indifferent to being observed. In the early morning, light crosses the retina and falls on the melanopsin cells. The signal moves along the retinohypothalamic tract, arrives at the suprachiasmatic nucleus, and meets the ventral core neurons waiting there. Glutamate and PACAP are released. The PER genes are briefly rapidly induced. The clock receives its correction, absorbs it into its ongoing oscillation, and continues. The molecular day recommences, all of the sleepiness that will gather through the coming afternoon and evening, the soft narrowing of attention, the way the body will begin to feel the pull of the horizontal, the slow closing of the world into something smaller and quieter and more interior. All of it begins here, in this adjustment, in the continued turning of a clock that has been running inside every human being who has ever lived, in every animal that has ever slept, in every organism complex enough to need one, for longer than there have been eyes to see the light that sets it. The loop turns. PRG proteins begin their slow accumulation in the warm interior of cells, no wider than a small fraction of a millimeter. The clock moves forward at the rate its chemistry allows, corrected by light, distributed across a network of 20,000 cells, firing in coordination, their signal flowing outward through the hypothalamus and into the rest of the brain, shaping in its passage the long, quiet arc of the day, the timing of alertness, and the timing of its release, the hour when the world begins to feel like somewhere it is right to rest. And somewhere in those first morning hours, when the correcting light has arrived, and the clock has received it, and the molecular day is fresh in its beginning, the suprachiasmatic nucleus puts forward its signal with the particular quality that belongs to waking, a firing pattern that supports the arousal systems downstream, a chemical output that holds the door of the day open, a quiet insistence that runs through the morning and into the afternoon. And then, slowly, as the loop continues its turn toward the far side of the cycle, begins to let that insistence go. The clock does not decide this. There is no decision. There is only chemistry cycling in cells smaller than a point of light, turning at the speed its proteins allow, carrying outward into the whole of the sleeping and waking body the single continuing signal that it was built to carry, the time. In the lateral hypothalamus, in the hour just after waking, there is a small population of neurons that hold the entire waking brain in their keeping. They number only in the tens of thousands. Each one sends out long, reaching axons that travel across the brain's full width, down into the brainstem, up into the cortex, sideways into the thalamus, forward into the forebrain, so that every structure involved in keeping a body alert and moving and attending to the world receives from this one small cluster a continuous and stabilizing signal. These are the orexin neurons. In the early morning, they are firing steadily, their projections reaching outward like the rigging of a ship held taut, the whole architecture of wakefulness kept upright by the tension they maintain. What they release is a neuropeptide, a short chain of amino acids that crosses the narrow fluid gap between cells and binds to receptors on the far side. There are two forms of it, slightly different in structure, both carrying the same broad message: remain awake, remain stable, do not slip. The cells that receive this signal are themselves the broadcasters of wakefulness, noradrenergic cells in the brainstem, histamine-releasing cells in the posterior hypothalamus, serotonin-producing cells along the brain's midline, and the cholinergic cells of the basal forebrain, whose output sustains the cortex in alert, fine-grained attention. The orexin neurons speak to all of them at once, and in the early hours of a waking day, all of them are listening. The orexin system is not simply one arousal signal among several. It functions more as a regulator of the entire set, a mechanism that prevents the waking and sleeping states from bleeding into one another at the wrong moment. Without it, the transition between states becomes unstable. The evidence for this comes from people in whom the orexin neurons have been lost, most of them destroyed by an autoimmune process that removes nearly all of them over a period of time. In those people, wakefulness does not simply feel lighter or less reliable, it collapses. Sleep intrudes without warning into the middle of standing, eating, speaking, the boundary between states, which in a healthy brain holds firm across the full arc of a day, becomes something that cannot be trusted. The role of the orexin system is to hold that boundary, to make the waking state something a body can rely on, and it does hold through the morning and into the late morning and through the first part of the afternoon. The neurons fire, the projections carry their signal, the arousal network holds its shape, but across all of those hours, something else is accumulating, building in the narrow fluid spaces between neurons, rising in the basal forebrain, spreading slowly through the regions the orexin system most depends upon. It is adenosine. It has been there since the first neuron fired that morning. It builds with every hour, with every synapse, with every small act of metabolic work the brain performs. And the orexin neurons, for all their reach and influence, carry receptors on their own membranes that bind to it. Across the afternoon, as adenosine rises, those receptors are progressively occupied, and slowly, by fractions that are too small to feel in any single moment, the orexin neurons begin to grow a little quieter. The locus coeruleus sits in the brainstem, small and bilateral, its two clusters separated by the fourth ventricle, and no larger together than a few cubic millimeters. Its neurons are the brain's primary source of norepinephrine, a neuromodulator that, when released across the cortex and thalamus, shifts neural processing toward a state of readiness, of narrow attentional focus, of heightened sensitivity to signals in the environment. In the first hours of a waking day, locus coeruleus neurons fire at a steady 1 to 4 times per second. This tonic rhythm is not dramatic. It does not spike and crash. It sustains continuously across hours of wakefulness, its output maintaining the cortex in a condition that is slightly more responsive, slightly more able to distinguish what matters from what does not. The relationship between this firing and the state of the body is precise. During wakefulness, the rhythm continues. During the slow, rolling waves of deep non-REM sleep, it progressively quiets. And during the strange, paralyzed dreaming of REM sleep, it falls almost entirely silent, as though the brain in that state has decided it no longer needs the kind of attention that norepinephrine supports, and has turned the signal off completely. The on-off precision of this pattern across the night was one of the first clues that different sleep stages are not simply different depths of the same process, but genuinely different states, each with its own neurochemical profile. The locus coeruleus marks the boundary between them. Adenosine acts on these cells through receptors on their membranes. The specific receptor type involved is a 1, the same class found broadly across the cortex and hippocampus, the same kind that, when bound, opens potassium channels and quiets the cell's electrical activity. As the day deepens and adenosine builds in the fluid surrounding the locus coeruleus, those A1 receptors are occupied in increasing numbers. The effect is not a sudden silencing. It is a slow reduction, the tonic firing rate declining by small fractions, the norepinephrine output across the cortex dropping in proportion, the cortex becoming slightly less responsive to each succeeding hour of the afternoon. The body does not notice a single moment in which this happens. What it notices, across many hours, is that the same tasks that felt easy in the morning now require slightly more effort, that distractions that were easily dismissed now carry slightly more weight, that the quality of attention available has, without announcement, become a little thinner. In the posterior hypothalamus, packed into a region called the tuberomammillary nucleus, are the brain's only histamine-producing neurons. In humans, they number roughly 64,000, a population smaller than many cell clusters in the brainstem, and their reach across the brain is enormous in proportion to their size. Histamine, released from these cells, acts on receptors in the cortex, thalamus, and hippocampus to promote and sustain wakefulness. The evidence for this is partly pharmacological. The drugs that block histamine receptors, the first-generation antihistamines developed decades ago, still found in many over-the-counter formulations, reliably produce drowsiness as a side effect because they are doing to the brain's histamine signaling precisely what the natural accumulation of sleep pressure does across an afternoon. The antihistamine does not generate sleep directly. It reduces the wakefulness signal, and what fills that space is the accumulated sleep pressure that was already present, waiting. The tuberomammillary neurons have a particular quality that distinguishes them from some other arousal nuclei. Their firing is wakefulness exclusive in an especially sharp way. During wakefulness, they are active. The moment sleep begins, not deep sleep, not a transition that takes minutes, but the beginning of sleep, they fall silent. The timing of this cessation is nearly absolute. No other nucleus in the arousal system has quite the same on-off precision. This quality makes them both a reliable marker of the state transition and a clue to the architecture of sleep itself, that the onset of sleep is not a gradual dimming of all signals simultaneously, but a structured collapse that involves different systems falling silent in a specific order, each one withdrawing its contribution to the alert state as the balance tips toward rest. Adenosine and histamine do not interact only through receptor occupancy at distant sites. The tuberomammillary neurons themselves receive input from the adenosine system, and the accumulation of sleep pressure across the day contributes to the progressive reduction of their activity during the late afternoon hours. By the early evening, the histamine signal that was steady and full in the morning is already diminished. The cortex that relied on it for a certain baseline of arousal is receiving less. The gap between the alertness of 9 in the morning and the alertness of 9 in the evening is built, in part, from the slow withdrawal of this signal from a population of 64,000 neurons in the hypothalamus that has been awake and firing since dawn and is now, hour by hour, releasing its hold. Along the brain's midline, arranged in clusters that run from the midbrain into the upper brain stem, the RAFE nuclei produce serotonin. The relationship between serotonin and sleep is not simple. Serotonin is not a sleep signal, and its story across a day does not move cleanly in one direction. But the dorsal and median RAFE neurons, like their counterparts in the locus coeruleus, fire tonically during active wakefulness, reduce their activity during drowsiness, quiet further during non-REM sleep, and fall nearly silent during REM sleep. This state-selective pattern places them firmly within the arousal network. Their output is part of what sustains the waking state, and its withdrawal is part of what allows sleep to begin. Serotonin has a relationship to another molecule that matters to the daily rhythm of alertness. The pineal gland, a small structure deep in the brain that responds to signals from the biological clock, uses serotonin as a biochemical precursor in its synthesis of melatonin. The two are chemically linked. Serotonin comes first in the pathway, and melatonin emerges from it in the darkness of the evening. This relationship means that the serotonergic system, which is active during the waking day and quiets toward evening, is feeding into the same transition that the rest of the arousal network is undergoing, contributing, indirectly, to the hormonal signal that will accompany sleep's arrival. The afternoon serotonin withdrawal is not experienced as a specific feeling. It does not announce itself. It is one thread among several in the slow unraveling of the arousal network across the hours between noon and nightfall. A thread that removed alone would not be noticed, but that removed alongside norepinephrine, alongside histamine, alongside the diminishing support of the orexin system, becomes part of a coherent and measurable shift in the quality of the waking state. The basal forebrain completes the picture. Its cholinergic neurons, concentrated in the nucleus basalis of Meinert and the surrounding substantia innominata, project widely across the cortex and are essential to the fine-grained quality of waking attention. When they fire, the cortex is desynchronized, its electrical activity dominated by fast, low-amplitude rhythms that correspond to active, engaged processing. When they quiet, the cortex begins to show slower rhythms, the emergence of alpha and theta waves that mark the border between attention and drift. And these neurons are precisely the population most sensitive to adenosine. The basal forebrain is the primary site of adenosine accumulation across a waking day. It is where the homeostatic sleep pressure is most concentrated, where it has been building since the morning's first waking moment, and where its effects on the arousal network are most directly expressed. In a cat, in an experiment conducted in the late 1990s, researchers placed small sampling probes into the basal forebrain and measured the adenosine concentration in the fluid there across periods of wakefulness and sleep. What they found was a steady hour-by-hour rise during waking and a gradual decline during sleep, a pattern that matched almost perfectly the behavioral and EEG markers of sleepiness and recovery. The basal forebrain was not just a site of adenase accumulation. It was the site most directly linked to the behavioral transition into sleep. Remove the adenosine from it, or block its receptors there, and sleep pressure failed to translate into sleep behavior. The accumulation was necessary, but it had to happen in this particular place, in the neighborhood of these particular cholinergic neurons, to produce the full expression of sleepiness. This is the mechanism behind the afternoon's quiet heaviness. Not one thing withdrawing, but 5 orexin, norepinephrine, histamine, serotonin, and acetylcholine, each diminishing in its own way, through its own receptor pathway, on its own schedule, and all of them declining together as the adenosine tide rises in the spaces between the cells of the basal forebrain. The felt experience of this, the way a book held easily at 9 in the morning, begins to feel heavy at 3 in the afternoon. The way a thought that assembled quickly in the early hours now takes longer to form. The way the edges of attention soften without a specific moment at which they did so is the subjective face of a process that is running simultaneously in 5 neurochemical systems across the full width of the brain. Viewed from outside, from some elevated vantage that could see the whole brain at once, the arousal network in the early morning would look like a lit structure, orexin projections reaching from the lateral hypothalamus to every corner, norepinephrine broadcasting from the brainstem, histamine spreading from its small posterior cluster, serotonin moving along the midline pathway, acetylcholine activating the cortex from below. Each system distinct in its chemistry and its anatomy, but all of them aligned in their output, all of them in their different chemical languages, saying the same thing: remain alert, remain awake, the world requires attention. By mid-afternoon, that same view would show something dimmer, not dark, not collapsed, but measurably, quantifiably less bright than it was at 8 in the morning. The orexin projections are carrying a signal whose amplitude has been reduced by the adenosine that has accumulated around the orexin neurons themselves. The locus coeruleus is firing a little more slowly. The tuberomammillary nucleus is approaching the threshold at which its neurons will fall silent. Not there yet, not in the mid-afternoon for most people, but closer than they were at noon. The Rafe nuclei are quieter than they were, and in the basal forebrain, the cholinergic neurons, whose output holds the cortex in alert desynchronization, are receiving more adenosine at their receptors with each passing hour, their potassium channels opening incrementally, their threshold for firing rising by small steps that are invisible individually but cumulative across the arc of a day. The cortex that those cholinergic neurons project into is not passive in this. As the cholinergic input diminishes, the cortex itself changes character. Its fast rhythms, the beta and gamma activity that mark engaged, attentive processing, begin to give way in short intervals to slower waves. Alpha appears at the back of the brain first, the rhythm of relaxed non-engagement. Then, in the late afternoon under high sleep pressure, brief intrusions of theta, the borderline rhythm between wakefulness and the first stage of sleep. These intrusions are not sleep. The person experiencing them is still awake, still able to respond to the environment. But the brain, for fractions of a second at a time, is producing rhythms that belong more to sleep than to waking. The homeostatic pressure is beginning to press through. This is the physiological substrate of the afternoon heaviness, not tiredness in the sense of muscular fatigue, though that may also be present, but something specifically neurological, something happening at the level of receptors and channels and membrane potentials in 5 interrelated systems that have been at work since morning. The heaviness is the shape of accumulated work. It is what a brain that has been firing and processing and attending for many hours looks like from the inside. There are people in whom one part of this system is missing. In narcolepsy, in which the orexin neurons have been lost, the withdrawal of the stabilizing signal does not happen gradually across an afternoon. The boundary fails entirely without the slow preparation that accumulated adenosine normally provides. Sleep intrudes abruptly in the middle of a sentence or a meal, without the long approach that an intact orexin system allows the other arousal nuclei to prepare for. The contrast between that abrupt failure and the normal day's slow progression is one of the clearest demonstrations of what the orexin system actually contributes. Not wakefulness itself, but the stability of the transition between states, the gradual and ordered nature of sleep's arrival. In a person whose sleep the previous night was short or broken, the orexin system faces the day with adenosine already elevated. The tide did not fully recede overnight. It begins the morning higher than it would after a full and restorative sleep, which means the orexin neurons are already at 8 in the morning operating under a mild adenosine load. Their firing is not significantly reduced. The A1 receptor occupancy is still relatively low, but the margin is smaller. The point in the afternoon at which the cumulative adenosine concentration begins to meaningfully suppress orexin firing arrives a little earlier. The point at which the locus coeruleus slows, the tuberomammillary neurons approach their silence threshold, the basal forebrain cholinergic neurons become sluggish. All of it arrives sooner than it would in a person who slept well. The progression of the afternoon follows the same path. It simply starts further along it. This is why a night of poor sleep does not simply make the following morning feel worse. It reshapes the whole arc of the following day, compressing the window of reliable alertness, moving the afternoon's decline earlier, making the evening's drowsiness arrive with less warning. The adenosine that was not cleared during the shortened sleep carries forward into the new day and shortens the distance between the morning's start and the afternoon's diminishment. The system is recording the incomplete recovery. It keeps its own account. The 5 components of the ascending arousal network do not decline at exactly the same rate or at exactly the same hours. The histamine signal, which is the most absolutely wakefulness selective, may begin to falter relatively early in the afternoon under high sleep pressure. The locus corymbloides, whose tonic firing rate is less absolutely linked to the moment-by-moment state of adenosine, may sustain its rhythm slightly longer. The orexin system, which is doing the additional work of stabilizing the other four, may show its adenosine-mediated reduction over the longest time course of all, its decline spread across the full afternoon rather than concentrated in a single window. The serotonergic raphe, sensitive to both adenosine and to the circadian clock signals that will strengthen through the evening, follows its own composite trajectory. What the listener experiences as a continuous, undifferentiated feeling of late afternoon heaviness is actually the composite of 5 declining signals, each on its own schedule, each mediated by its own receptor mechanism, adding their reductions together into something that arrives at consciousness as a single smooth sensation. The brain does not label these separately. It does not distinguish between the norepinephrine component and the histamine component of its growing drowsiness. It receives the aggregate, and the aggregate feels like one thing, a softening, a slowing, a gentle lessening of the will to stay sharp. The adenosine that drives all of this is not, itself, a signal that the brain interprets as pain or distress. It does not announce itself with discomfort. It speaks to the arousal network through receptors that, when occupied, simply open potassium channels and lower the probability that a given neuron will fire. The neuron does not report that it is being inhibited. It simply fires less often, and the downstream regions that relied on its output receive less of the signal they had been depending on. The experience that emerges from this cellular level reduction, the soft spreading heaviness of late afternoon, is one of the brain's most precisely constructed signals. It does not say danger. It does not say something is wrong. It says, in the body's most fundamental chemical language, that the work of the day has been long and real, and that the conditions for rest are approaching. The orexin neurons in the lateral hypothalamus, by the time the evening arrives, are working against a rising tide. The adenosine that has been building since dawn has reached concentrations that are, across much of the basal forebrain and surrounding tissue, high enough to significantly reduce the orexin neurons' firing rate. The projections they send to the locus coeruleus, to the raphe nuclei, to the tuberomammillary nucleus, to the basal forebrain itself, all of them are carrying a signal that has been reduced in strength. The distant nuclei, already diminished by their own direct adenosine exposure, are receiving less support from the orexin system at the very time they need more of it. The two reductions compound each other. A single orexin neuron in this condition, viewed closely, shows the history of the day on its membrane. Its A1 receptors are occupied. The potassium channels they control are open at a higher baseline rate than they were in the morning. The neuron's resting membrane potential has shifted slightly, moved a fraction of a millivolt further from the threshold at which it would fire. It is still active. It is still sending its signal, but the signal is quieter than it was, and the quietness has been accumulating across 12 or 14 hours of wakefulness with the slow fidelity of a record that cannot be falsified. It is worth sitting with that image, not dramatically, not with urgency, but with the particular attention that this kind of precision deserves. Inside a structure that could be held in the palm of a hand, in a cluster of cells visible only under magnification, a neuron is carrying the full weight of the day's wakefulness in the state of its potassium channels. The molecule responsible is smaller than a wavelength of visible light. The channel it controls is a protein pore spanning a membrane measured in billionths of a meter, and yet the aggregate of all of these small molecular events in the orexin neurons, in the locus coeruleus, in the tuberomammillary nucleus, in the raphe, in the basal forebrain, is the afternoon. It is the specific quality of the light on the ceiling at 4 o'clock, the way the afternoon holds something heavier than the morning did, the slight softening at the edges of thought that arrives without being invited and is not, for all its quietness, unwelcome. The tuberomammillary nucleus approaches its transition. Its neurons are not yet silent. Silence comes at the moment of sleep, with a precision that has no gradual middle, but they are approaching the threshold at which the balance between their inputs will tip. The orexin signal that sustains them is diminished. The adenosine pressure on their own membranes has been building. The circadian alerting signal that counterbalances the homeostatic pressure through the afternoon is beginning in the early evening hours to decline. When all three of these conditions align, when the orexin input is reduced, the adenosine load is high, and the circadian alerting signal has begun its evening fall. The tuberomammillary neurons will fall silent with the sudden completeness that marks them as one of the most state-specific populations in the arousal system. Not a fade, a cessation. The locus coeruleus is already quieter than it was at noon. Its tonic 1 to 4 hertz rhythm, which in the morning was steady and unhurried, has declined. The norepinephrine it has been broadcasting across the cortex since waking is arriving in smaller amounts. The cortex is not yet showing the slow delta waves of sleep, but its responses to incoming signals, the small electrical events that mark attention being paid, information being processed, are slightly slower to form and slightly lower in amplitude than they were. The difference is not measurable in single moments. Across the arc of the day, it accumulates into something that is both chemically real and experientially familiar. The basal forebrain's cholinergic neurons are quieter than they have been at any point since morning. The cortex they project to, deprived of the full cholinergic input that sustained its fast rhythms, is showing more alpha, more brief theta, a slight but measurable shift in its electrical character toward the patterns that precede sleep. This shift is not itself sleep. A person in this condition is still awake, still responsive, still capable of sustained thought, but the brain's capacity to resist the next step, the actual transition into sleep, has been reduced, hour by hour across the full length of the day, by the accumulated adenosine that has been recording its every firing minute since the morning's first light. The raphe neurons along the midline have reduced their output. The serotonin that moved outward from them across the morning and into the afternoon is by the early evening being released at a lower rate. The pineal gland's melatonin synthesis, which depends in part on serotonin as a precursor, is beginning as the light of the evening changes and the biological clock releases its suppressive hold to rise. The two systems are handing off, one diminishing as the other begins. The transition is chemical, it is gradual, it has been prepared for across the entire length of the day, across the late evening. The 5 components of the ascending arousal network, orexin, norepinephrine, histamine, serotonin, acetylcholine, have all diminished from their morning levels. None of them has reached zero. The system is not yet in sleep mode. It is at the lowest point of the waking day, the trough that the homeostatic pressure has been carving toward since the first hour after waking. The adenosine that drove this carving is present in concentrations that represent the full accumulated work of a long day. Every receptor it occupies, every potassium channel it has opened, every small reduction in firing rate it has produced across 5 neurochemical populations. All of it is the molecular record of the day's duration. The orexin neurons, still active, are sustaining the waking state against the full weight of that record. They will continue to do so until the moment the circadian alerting signal drops far enough and the homeostatic pressure rises high enough that the balance tips, until the combined withdrawal of circadian support and the accumulated adenosine load exceed the orexin system's capacity to hold the waking state stable. When that moment arrives, it arrives as sleep, not as a gradual fading, but as a transition, one that will have been approached so slowly across so many hours that its arrival, when it finally comes, feels less like a crossing than like a settling, a coming to rest at a destination that the day's whole chemistry was oriented toward from its first waking moment. The 5 systems that held the brain in wakefulness, that fired steadily through the morning, carried the weight of the afternoon, sustained their signals through the hours of accumulating pressure, will all, in the course of that transition, fall quiet in their own ways and their own orders. The tuberomammillary nucleus will fall silent first, and most absolutely, the locus coeruleus will quiet by degrees. The raphe neurons will reduce further still. The orexin neurons will slow to their lowest waking rate, and then, with sleep's arrival, settle into the diminished firing that accompanies the night's first deep stage. And the basal forebrain's cholinergic neurons, the ones whose quieting allows the cortex to release its fast rhythms and accept the slow ones, will complete what the adenosine has been building toward all day. A stillness that is not absence, but recovery, not failure, but the faithful accounting of everything the day required. The ascending arousal network in the late evening is not a system in distress. It is a system that has done what it was built to do, held the waking state stable across many hours, sustained attention through the morning and the afternoon, adapted to the rising tide of adenosine without breaking, and now, with the day's work measured and recorded in the concentration of a molecule smaller than any sense can detect, is releasing its hold on wakefulness with the same steadiness with which it maintained it. There is no drama in this. There is only the quiet precision of 5 neurochemical systems declining together, their collective output diminished to the point where the transition into sleep becomes the path of least resistance. The orexin projections grow still. The norepinephrine signal, which began the day as a steady distributed hum across the whole cortex, has grown faint. The tuberomammillary neurons are on the edge of their characteristic silence. The raphe neurons, already quiet, are quieter still, and in the basal forebrain, in the neighborhood of the neurons that have been accumulating adenosine since the morning, the slow tide that has been rising all day has reached the level at which the system can no longer hold what it has been holding. The day's account is settled. The record is complete. And what comes next? What is already arriving, already beginning in the slow electrical changes moving through the cortex, is exactly what the day's chemistry has been building toward from the first hour of its beginning, moving toward it with the unhurried certainty of something that has always known where it was going and has been going there, gently, all along. A small screen glows in a quiet room. On it, a single horizontal line moves from left to right, interrupted every few seconds by a brief flash of light. A finger rests on a response button. The task is simple. See the light. Press the button. The line records how long it takes. Each trial is identical. Each hour that passes without sleep makes it by a small but measurable amount harder. The psychomotor vigilance task does not ask much of the person sitting before it. It asks only that they remain present, that the gap between signal and response stay narrow, and for the first 12 or 13 hours of a waking day, Most people hold that gap close. The line stays relatively flat. The lapses are rare. The system is doing what it was built to do, sustaining a kind of readiness, moment after moment, across the long arc of a day in which nothing catastrophic has happened and nothing dramatic has been demanded. But the flatness is not permanence. It is held in place by a balance of forces, and those forces are not equal in strength at every hour. After 16 hours awake, the curve begins to tilt, not sharply, not all at once. The change arrives the way most biological changes arrive, as a slow divergence from what had seemed stable, a drift that is invisible at any single moment and unmistakable across the span of an hour. Reaction times that average 250 milliseconds early in the morning begin to approach 300, then 350. Lapses, moments when the response fails to arrive at all within a fixed window, begin appearing with greater frequency, first singly, then in clusters, then as a pattern that cannot be explained by inattention or boredom alone. Something structural is changing inside the brain, something that has been changing all day, and the task is now sensitive enough to detect it. The change it is detecting has been accumulating since the moment of waking. It has been building in the gap between neurons, rising with each hour of metabolic work, carried in the slow tide of a small molecule that the brain produces as an inevitable consequence of being awake. Every neuron that fires, every synapse that transmits, every process of maintenance and signaling and repair that runs through the cortex in the brainstem and the forebrain, all of it leaves a trace in the extracellular fluid, a trace that the brain itself can read. And across the full length of a waking day, that trace deepens. It becomes denser. It presses more firmly against the receptors that respond to it, opening channels and quieting cells, and dimming the bright distributed network that has been sustaining alertness since before the first memory of the morning. What the psychomotor vigilance task measures is the visible surface of a process that runs far beneath behavior. The slowing reaction time is not itself the mechanism. It is the reflection of the mechanism, the shadow of what is happening at the level of cell and molecule, cast outward into something that can be clocked in milliseconds and plotted on a curve. And that curve is not the same in everyone, and not the same on every day, and not fixed at any given hour. It is shaped by the night that preceded it, and the hours of wakefulness that have followed, by how deeply the brain slept, and for how long, and whether the slow, deep waves of early sleep fully discharged the load that had built through the day before. By the time of day, which exerts its own independent pressure on the level of alertness that remains available. By each of these things pressing on the system simultaneously, their effects combined into the single moving number that the task records, the gap in milliseconds between signal and response. Research following groups of people through extended periods of restricted sleep found something in that curve that was not easy to predict. A group sleeping 6 hours a night for 2 weeks did not feel as impaired as they would have expected. The subjective sense of sleepiness, the felt weight of fatigue, the self-reported difficulty staying alert, plateaued after a few days and did not continue climbing, but the reaction time kept rising, the lapses kept accumulating, the task kept detecting what the person could no longer feel. After 14 nights of shortened sleep, the measured impairment was equivalent to what would be seen in someone who had gone a full 24 hours without any sleep at all, while the person themselves reported only moderate tiredness. The kind of tiredness that seemed manageable, the kind that did not feel like emergency. This divergence between how impaired a person is and how impaired they believe themselves to be has a particular quality that bears sitting with. It is not distraction or inaccuracy or a deliberate underestimate. It is a genuine recalibration of the baseline. When performance declines slowly across days, the brain adjusts its sense of what normal feels like. The new worse performance becomes the reference point. The gap between current function and optimal function becomes invisible because optimal function is no longer a felt memory, only an absent one. The person is not lying about how they feel. They are telling the truth about a perception that has drifted away from what the measurement shows, the two lines separating on a graph with the quiet patience of a slow tide. The Karolinska Sleepiness Scale offers a different lens onto the same separation. It is a simple instrument, 9 points, from fully alert to fighting sleep, and when administered alongside the task that measures reaction time, it tracks alongside it reasonably well through the first 16 or 17 hours of waking. Someone who rates themselves a 5 or 6 on the scale is indeed producing more lapses than someone who rates themselves a 2. The correlation holds. The self-report reflects the physiology, at least for a while. But somewhere after 20 hours of continuous wakefulness, the relationship begins to loosen. Subjective ratings plateau or even edge slightly downward. The person reporting that they feel a little better, a second wind arriving, a sense of stabilization. And the task recording continues its slow climb. The lines move apart. The felt and the measured become across the final hours of an extended night, something close to strangers. The wave of alpha and theta activity in the waking EEG tells a quieter version of the same story. Alpha waves, those slow oscillations between 8 and 12 cycles per second that appear in the resting waking brain when the eyes are closed, begin to intrude into periods of apparent alertness as the day progresses and sleep pressure mounts. Theta waves, slower still, between 4 and 8 cycles per second, characteristic of drowsiness and the lightest edges of sleep, begin appearing in the frontal lobes during bouts of attempted wakefulness, brief intrusions of a sleep-like pattern into a brain that has not yet surrendered consciousness. An electrode placed over the frontal cortex of someone sitting upright, eyes open, engaged in a task, will begin to pick up these waves more frequently in the late afternoon and evening than it would have in the mid-morning. The measure is not behavioral, not self-reported. It is the direct electrical signature of a brain whose balance has begun to tip. Inside that tipping, something is happening that no electrode can directly record, but that every waking nervous system enacts every single day. The ascending arousal network, that distributed neurochemically diverse system, sustained by neurons in the brainstem and hypothalamus and basal forebrain, is not a fixed structure. It is a dynamic one. It is sensitive to what is built up around it, and what is built up around it across a day of unbroken wakefulness is a rising concentration of a molecule that those neurons' own receptors are designed to detect. Every hour they spend firing adds to the pool. Every neurotransmitter released, every process of synaptic maintenance completed, every cascade of energetic work performed by the metabolically active cells of the cortex and forebrain contributes, through the sequential breakdown of adenosine triphosphate, to the slow extracellular accumulation of adenosine. And adenosine presses on those neurons. It opens channels in their membranes. It shifts their electrical state towards silence. Gradually, across hours, the great arousal network grows a little quieter. Not all at once, not in any single, legible moment, but measurably, cumulatively, in a direction that can only go one way until sleep arrives and begins to clear the accumulated load. The cortisol awakening response reaches into this picture from a different angle, not from the homeostatic side, not from the accumulation of sleep pressure, but from the circadian machinery that runs beneath and alongside it. In the first 15 to 45 minutes after waking, before the first meal or movement, before the day has made any demand at all, the level of cortisol in the blood rises sharply. It may increase by 50 to 100% above the level present during sleep, a brief and reliable surge that appears every morning in every healthy adult with a functioning circadian system. The surge is driven by the suprachiasmatic nucleus, the brain's circadian pacemaker, activating the chain of signaling that runs from hypothalamus to pituitary to adrenal cortex. It is not a response to stress. It is a preparation for the day, a mobilization of the body's alerting chemistry, timed to the expected hour of waking, set in motion by a clock that has been running through the night in the dark of the hypothalamus, counting time without any light to tell it what hour it was. The amplitude of that morning cortisol surge is not fixed. It varies from morning to morning, and part of what shapes it is the quality and quantity of the sleep that preceded it. A night of deep, architecturally complete sleep, in which the slow wave activity of early non-REM discharge, the accumulated homeostatic load, and the later cycles of lighter sleep and REM complete their own slower work, produces a cortisol awakening response of fuller amplitude than a night of shortened or fragmented sleep does. The surge is smaller. The mobilization is less complete. The day begins from a shallower platform, and the tools available for sustaining alertness across that day start already partly depleted. The sleepiness that appears by early afternoon on a day following poor sleep is not simply more of the same process. It is the same process running on a reduced budget, the homeostatic load not fully discharged, the circadian alerting signal not fully supported by the hormonal preparation that was supposed to accompany it. By mid-morning, the brain is in one of its most alert states of the entire day. The cortisol surge has peaked and begun its descent, but its effects on alertness persist. The circadian alerting signal from the suprachiasmatic nucleus is climbing toward the broad plateau of its daytime maximum. Core body temperature, which tracks the circadian alerting signal with a delay of several hours, is still rising. The homeostatic sleep pressure, accumulated since waking, is present but not yet dominant. The two processes that shape sleep timing are in a particular relationship at this hour, one climbing, the other growing, but their opposition still clearly resolved in favor of wakefulness. The brain can sustain attention, it can hold information in mind, it can return to a disrupted task and find its place again without effort. This is not a coincidence of circumstance. It is the product of a system working at close to its full capacity, both of its major drives temporarily aligned on the same side. The alignment does not last. At some point in the early afternoon, the specific hour varying with chronotype and with the prior night of sleep, but falling for most adults between 1 and 3 in the afternoon, something in the balance shifts. Core body temperature, which has been rising since its low point in the pre-dawn hours, pauses in its climb. It does not fall sharply. It softens very slightly before continuing upward again. The circadian alerting signal dips in a secondary trough. The homeostatic sleep pressure, still climbing since morning, reaches an amount that begins to be felt more clearly. And the EEG, if one were recording it, would show a brief increase in theta power in the frontal and central regions, not the deep theta of full drowsiness, but a quiet thickening of the slow oscillations that signal a brain whose load is beginning to press on its available resources. This is the post-lunch dip. The name is misleading because it is not caused by lunch. People who eat nothing through the morning still show it. People who eat a large breakfast and skip lunch still show it. It appears in populations that do not follow a midday meal tradition, and it appears in laboratory studies in which food timing has been carefully controlled and its effects isolated. The dip in alertness that settles over the early afternoon is a genuine feature of the circadian architecture, a secondary wave in the pattern of the body's daily oscillation, and has been written into the biological day of every animal that follows a light-dark cycle long enough to have developed an internal clock to track it. In the same early afternoon window, a short period of sleep does something that would not be possible at most other times of day. A nap of 20 or 30 minutes taken during this window, after the dip has arrived but before the circadian alerting signal resumes its climb toward the evening maximum, reduces theta power in the EEG for the hours that follow, restores performance on tasks that measure sustained attention, and does so without carrying enough sleep pressure, discharge to meaningfully delay the arrival of sleep that night. The window for this is real and relatively narrow. A nap taken too late in the afternoon, or one that extends past 30 minutes into deeper slow-wave sleep, will reduce the homeostatic load enough to push back the natural onset of nighttime sleep, shorten slow-wave sleep duration in the first cycle, or both. But a nap timed to the dip, brief and carefully bounded, is something the biology of the afternoon actively makes room for. There is something restful in the knowledge that the impulse toward afternoon rest, felt across cultures, observed in populations separated by geography and custom and daily schedule, is not a weakness or an indulgence. It is a feature of the circadian curve. The secondary trough is real. The slight fall in core body temperature is real and measurable. The rise in theta power is real. What the body is registering in those hours is not the accumulated fatigue of a morning's work or the weight of a meal digested, but a genuine dip in the underlying wave that carries alertness across the day. A wave that was set in motion not this morning, but over the course of a lifetime of daily cycles, calibrated across years of experience to the specific light-dark pattern of the place and season in which a particular life is lived. The late afternoon carries its own particular quality. For many adults, the hours between 4 and 7 in the evening hold a plateau of alertness that can feel for a time almost like a second morning. Core body temperature, still near its daily maximum, supports fast processing and physical coordination. The circadian alerting signal is at or near its peak. The homeostatic sleep pressure, though considerably higher than it was at noon, is still partially offset by the strength of the circadian drive. This is the wake maintenance zone, a period in which the body, even carrying a full day of accumulated sleep pressure, actively resists the transition to sleep. The circadian system in the late afternoon is not neutral. It is pushing back against the homeostatic load, holding the waking state open with a signal that is near its daily maximum, precisely at the time of day when fatigue would otherwise threaten to overwhelm it. The wake maintenance zone is not experienced as effortlessness. It does not feel like morning alertness, which arrives with the cortisol surge and the low homeostatic load of a freshly completed night. It has a different quality, the sense of being held awake by something rather than simply being awake. A slight tension in the alertness that was not present at 9 in the morning, but it is real, and its reality has been demonstrated in studies where people were kept on extended days of 28 hours, too long for any internal clock running on a near 24-hour cycle to track, and asked to try to sleep at evenly distributed intervals across the artificial day. The times when they could fall asleep most easily were clearly predictable from the circadian phase, not from the amount of prior wakefulness. And the times when they found it hardest to sleep despite carrying more than 16 hours of continuous wakefulness, fell precisely in the late afternoon and early evening when the circadian alerting signal was at its strongest. That resistance is not permanent. Nothing in the biological day is permanent. The wave that has been holding wakefulness open against the homeostatic tide peaks in the early evening and then begins its fall. And the fall, when it begins, is not gradual. The change in the circadian alerting signal in the hours between 8 and 11 at night has a steepness that the morning rise does not quite match. The body moves quickly, in biological terms, from the peak of its daily alerting drive toward a nadir that will not arrive until the early hours of the next morning. And it is in the context of this falling signal that the accumulated homeostatic sleep pressure, which has been building since waking, held partially in check by the circadian system through the late afternoon, becomes the dominant force. The pineal gland, a small structure nestled in the center of the brain, begins releasing melatonin into the bloodstream roughly 1 to 2 hours before the body's habitual sleep time. The release is not triggered by darkness alone, though darkness is necessary for it to proceed. It is timed by the suprachiasmatic nucleus, which has been signaling the pineal gland in rhythm with its own internal clock all day, and which begins shifting that signal in the hours before sleep to permit melatonin synthesis and release. The melatonin that enters the circulation does not cause sleep directly. It is a signal of darkness, a hormonal marker of the biological night, read by receptors in the SCN itself and by tissues throughout the body. It narrows the window during which the circadian alerting signal would otherwise hold wakefulness open, allowing the homeostatic pressure, which at this hour is large, to meet less resistance. The core body temperature that has been near its daily maximum through the late afternoon and early evening now begins to fall. The fall is caused partly by the opening of blood vessels in the hands and feet, a dissipation of heat through the skin's surface that is orchestrated by the same circadian system that has been running all day. The hands and feet warm as core temperature drops, an exchange of thermal energy that has been observed so reliably that the ratio of distal to proximal skin temperature has been used as a measure of circadian phase in research settings. The warmth in the hands and feet, the slight cooling at the body's core, these are not symptoms of fatigue. They are the circadian system preparing the body for the specific physiological conditions of sleep, when core temperature must be a degree or more lower than its waking maximum, when the metabolic rate drops, when the work of restoration begins. In the brain, something is quieting. The locus coroploius, which has been sending norepinephrine across the cortex since morning, reduces its firing rate. Its neurons, which were discharging at 1 to 4 times per second during the most alert periods of the day, fire less and less frequently in the hour before sleep. The tuberomammillary nucleus, which has been producing histamine exclusively during wakefulness, a nucleus that fires with a precision that is almost binary in its on-off character, grows silent. The dorsal and median raphe nuclei, sources of serotonin throughout the day, reduce their output as the evening deepens. And the orexin neurons of the lateral hypothalamus, the long-reaching, stabilizing system that has been holding the ascending arousal network together against all perturbation through the hours of the day, find their activity progressively suppressed as the adenosine that has accumulated against their receptors tips the balance. Each of these changes is small in isolation. None of them is by itself sufficient to produce sleep. The transition into sleep is not the failure of any single system, but the convergence of many small shifts in the same direction, each arriving from a different mechanism, each building on what the others have already changed. The homeostatic pressure is large and has been large for hours. The The circadian alerting signal is falling, melatonin is circulating, core body temperature is dropping, the ascending arousal network is growing quiet across 5 different neurochemical populations simultaneously. All of these are happening at once in the same hour in a convergence that has been organized across the full length of the day, not planned, not coordinated by any single point of control, but produced by independent systems that evolved across deep time to arrive at the same conclusion at the same hour. Sleep, when it comes, does not arrive like a door closing. It arrives like a light dimming, a slow reduction in the waking brain's own signal, a gradual softening of the electroencephalographic pattern from the mixed fast frequencies of alert wakefulness through the slower alpha of drowsiness and closed eyes into the first slow oscillations of stage 1 sleep, where the boundary between waking and sleep becomes difficult to name. In the seconds and minutes around sleep onset, the distinction between the two states loses its sharpness. A person can be woken from early stage 1 sleep and report that they were still awake. The EEG disagrees. The thalamus, already beginning to shift its firing pattern toward the rhythmic bursting that will eventually generate sleep spindles, does not share the cortex's opinion of its own wakefulness. Sleep has already begun in some parts of the brain, while others continue to register something like awareness. The first deep sleep of the night absorbs the homeostatic load with a thoroughness that no other stage matches. Slow wave sleep, the delta oscillations of stage 3, the high amplitude waves between half a cycle and 4 cycles per second that can be measured with an electrode taped to the scalp, carries within its power spectrum the direct signature of process S discharge. The deeper and more sustained the slow wave activity in the first cycle, the higher the homeostatic load that preceded sleep. A night of total sleep deprivation, followed by recovery sleep, produces first cycle slow wave activity of exceptional power and density. The homeostatic system drawing down the largest accumulated balance it is capable of carrying, paying back what it owes with the most efficient means available to it. A normal night of adequate prior sleep produces less dramatic slow wave activity, not because the first cycle is less important, but because the debt being discharged is smaller, the prior night having already cleared most of the load. Within a few minutes of slow wave sleep onset, the processes of neural restoration begin. Synaptic strengths that have been incrementally potentiated through the day's learning and activity undergo a systematic downscaling. Metabolic waste products that have built up in the interstitial space, including adenosine itself, though its clearance is not purely the work of sleep, begin to clear. The glymphatic system, driven by the slow wave synchronized pulsations of cerebrospinal fluid that flow most powerfully during deep sleep, moves through the spaces around neurons and carries away the molecular debris of a day's work. The cholinergic neurons of the basal forebrain, which have been quieting for hours as adenosine rose around them, are now receiving less of the molecule that has been pressing against their receptors all day. The tide, in sleep, begins to fall, and when it has fallen far enough, when the slow wave sleep of the first 90-minute cycle has discharged enough of the accumulated homeostatic load, when the balance has shifted from deep delta toward the lighter sleep of stage 2, when the first cycle approaches its close and the brain prepares to enter the strange physiology of REM, the person lying in the dark is no longer the same person who sat before the response task in the morning and held a 250 millisecond gap between signal and response. They are in a different state entirely. Their cortex is being reorganized by sleep, their memory consolidated, their emotional processing rebalanced. The homeostatic pressure that was large is becoming small. The circadian system, which will not begin its alerting rise again until the small hours of the morning, sits at its nadir. The entire system is in its deepest biological night, doing the particular work that only night allows. The psychomotor vigilance task, if administered now, would find no one to administer to. The person is elsewhere, inside the deep architectures of slow-wave sleep, moving through the episodic landscapes of REM, returning briefly toward waking in the lighter intervals between cycles, and then descending again. The reaction time has been set aside. The task will be waiting in the morning, and in the morning the gap will be narrow again, the lapses rare, the balance temporarily restored. Not because anything has been fixed, but because the system has done what it was built to do in the hours that were always going to be needed. The homeostatic load has been discharged. The circadian tide will turn again toward morning. There is something in the regularity of this that holds a kind of weight, not a heavy weight, but a steady one, like the weight of a tide that has been running in and out of the same shore for longer than there have been names for tides. The body measures its own wakefulness with molecular precision. It records every hour of effort in the rising chemistry of the extracellular fluid and carries that record forward through the day. It counts the hours against the turning of its internal clock. It prepares the transition to sleep across the full afternoon and evening, not in a single instant, but in a slow convergence of independent processes arriving together. And then it sleeps, and in sleeping, it clears the record, not permanently. The next waking will begin the accumulation again, the same molecule rising, the same neurons quieting, the same curve climbing toward its next inevitable peak. But for the hours of the night, the record is held in abeyance. The tide is out. The specific molecule that has been rising all day, that small purine nucleoside, structurally simple, produced as a byproduct of the brain's own energetic work, has no preference for sleep over wakefulness. It carries no intention. It simply accumulates where neurons have been active and binds to the receptors built to receive it, and the cells that carry those receptors respond by growing quieter, and the network those cells compose responds by growing quieter still. That this process, iterated across billions of cells and dozens of hours, produces the felt experience of sleepiness, the heaviness behind the eyes, the slowing of thought, the increasing difficulty of holding the mind to any single thing, is not something that requires a special explanation. It is the direct and faithful consequence of a system doing exactly what it was designed to do, measured now in milliseconds and plotted on a curve, felt now as the slow dissolution of the day's last alertness into the beginning of something darker and deeper and still. The room around the sleeping person has not changed. The same walls, the same dark, the same quiet. But inside the skull resting on the pillow, the chemistry has shifted. The tide is low. The delta waves move slowly through the quiet cortex, unhurried, deep, persistent. Outside the window, if there is one, the night continues at its own pace. The stars in their courses, the slow turn of the earth, the darkness that will give way to light again in a number of hours that the suprachiasmatic nucleus has already anticipated and built its schedule around.